Hello dears, now we will be learning about polymerase chain reaction or PCR. Coming to the introduction, PCR it is a method of in vitro cloning and it allows the amplification of specific DNA fragments in vitro, that is in lab conditions, through several cycles of enzymatic DNA synthesis. It is a repeated cycles of enzymatic DNA synthesis. That means DNA synthesis with the help of several enzymes, and it allows the amplification or scaling up of target DNA molecules. It is invented by Carey Mullis in 1984 and it is the most popular and widely used technique in all fields of biological studies. And this PCR technique, it is simple, sensitive, specific, reliable and fast. The equipment used for carrying out this PCR is known as thermal cycler. So PCR is a process and the equipment we use for this PCR process is known as thermal cycler. So the PCR is a process in which DNA sequence is artificially amplified by repeated cycles of replication and strand separation, generating thousands to millions of copies of a particular DNA sequence. It is a process or technique which we use to amplify or increase the number of a particular DNA sequence, and it includes various cycles, repeated cycles of DNA replication and strand separation. Coming to the definition of this PCR, it is a genetic technique that occurs in vitro which allows the enzymatic synthesis of large quantities of a target region of DNA. The DNA is synthesized in the same manner as that seen in vivo using a DNA polymerase enzyme. That means in the PCR technique, the method of synthesizing DNA is exactly the same method in which it occurs in vivo. In vivo means in the living cells. The exact mechanism is repeated in the lab conditions in PCR. Coming to the PCR principle and components. The PCR amplifies a specific region of DNA strand. Most PCR methods typically amplify DNA fragments of between 0.1 and 10 kilo base pair. The components and reagents used for this PCR are: first we should have a target DNA or DNA template, and we should have two primers, one which is a forward primer and one reverse primer for the DNA template. Then the enzyme Taq polymerase, that is DNA polymerase enzyme. Then deoxynucleoside triphosphase, that is DNTPs. So we should have these components. That means first one, it is the gene of interest, the gene or the DNA fragment to be amplified, and we should have two primers, that is reverse and forward primers for the target DNA. Then we should have a Taq polymerase enzyme, which is a DNA polymerase enzyme, and then DNTPs. The other reagents are buffer solution, divalent cations and monovalent cations. The main steps of PCR is denaturation, annealing and extension. Denaturation, in denaturation, the double-stranded structure of the target DNA is denatured to form two single strands. And then in annealing step, the primers, forward and reverse primers, anneal to the target DNA fragment. Then extension, the third step involves the extension of the primers to form a complete DNA segment. And while we do this PCR in lab condition, we have 3 more steps before and after these 3 main steps. That means first we have an initialization step, and after denaturation, annealing and extension, we have final elongation and final hold step in PCR when we do in lab conditions. Coming to the procedure. The PCR technique copies the target DNA by performing repeated cycles, each containing the following step. First step, it is initialization step, which consists of heating the reaction to a temperature of 94 to 96 degrees Celsius for 1 to 9 minutes. This is just a warming up step for this PCR by heating the reaction system to 94 to 96 degrees Celsius for 1 to 9 minutes. And the second step comes the denaturation or melting step. It is the main step we discussed earlier. It is the first regular cycling event and consists of heating the reaction to 94 to 98 degrees Celsius for 20 to 30 seconds. It causes melting of the DNA template by disrupting the hydrogen bonds between the complementary bases, yielding single-stranded DNA molecules. As you all know, the denaturation step or melting step involves the giving of high temperatures so that the hydrogen bond between the nitrogenous bases the hydrogen bonds between the bases disrupts and thus separates the double-stranded DNA into 2 single strands, 2 separate strands. For confirming this denaturation to 100%, we are giving this initialization step. Then the next step, it is annealing step. The reaction temperature is lowered to 50 to 65 degrees Celsius for 20 to 40 seconds, allowing the annealing of the primers to the single-stranded DNA templates. In annealing step, the temperature is lowered. It is cooled. In this figure, you can see the bluish color indicates the lower temperature for the reaction, and in the previous slide we saw the picture. It is having a background of red color, which indicates the high temperature condition. So in annealing step, the temperature is lowered to 50 to 65 degrees Celsius, so that we are giving the primers a chance to bind to the DNA template by hydrogen bonding. Okay, then this temperature must be low enough to allow for the hybridization of the primer to the strand, but high enough for the hybridization to be specific. That is, primer should only bind to a perfectly complementary part of the template. So in this step, the temperature should not be lowered very, but enough temperature should be there for the primer to bind to the DNA template specifically, only to the complementary base pair. Okay, so here after annealing step, we can see that the primers forward and reverse primers has been attached to the DNA template. Then the last main step of the PCR, it is the elongation step or extension step. The temperature at this step depends on the DNA polymerase used. Taq polymerase has its optimum temperature at 75 to 80 degrees Celsius, and commonly a temperature of 72 degrees Celsius is used with this enzyme. So the temperature of this elongation step depends on the DNA polymerase we are using, and if we are using the Taq polymerase, the optimum temperature is 75 to 80 degrees Celsius, and usually 72 degrees Celsius is used. Okay. At this step, the DNA polymerase synthesizes a new DNA strand complementary to the DNA template strand by adding DNTPs that are complementary to the template in 5 to 3' direction. So the extension of or the elongation of the primers occurs in this step. So the DNA polymerase or Taq polymerase helps in the addition of DNTPs that are complementary to the base pairs in the template strand. So it helps in adding up the DNTPs to the template strand in 5' to 3' direction. Always the elongation and the DNA replication, everything done is occurring in 5' to 3' direction. The process of denaturation, annealing, and elongation constitute one cycle. These 3 steps involves one cycle. Multiple cycles are required to amplify DNA. So, how if we need the aim of doing PCR is to amplify or make several thousand copies of DNA. So, after one cycle of PCR, that is denaturation, annealing, and elongation, the number of copies is doubled. So we have to repeat these cycles again and again for getting thousands of copies. So the formula used to calculate the number of DNA copies formed after a given number of cycles repeated is 2 raised to n number of cycles, where n is the number of cycles. So if we are doing 30 cycles, we are repeating 30 cycles of PCR, we can calculate the amount of DNA we are getting after the 30 cycles by calculating 2 raised to 30. We will be getting that number of DNA molecules after PCR. Then in PCR technique, the main 3 steps are repeated in a cycle. The initialization, this final elongation and final hold will be only for once. So initialization is the first step, and the second step denaturation, third step annealing, fourth step extension. This denaturation, annealing, and extension steps are repeated in the cycles. That means if we are doing 30 cycles of experiment, then 30 times these 3 steps are repeated, not initialization and final elongation. Only these 3 steps are repeated. So coming to the after 30 cycles of repetition of this PCR cycle, finally in the last cycle we will be going to this final elongation. This is a single step, not a repeating step. This single step is performed at a temperature of 70 to 74 degrees Celsius for 5 to 15 minutes after the last PCR cycle to ensure that any remaining single-stranded DNA is fully extended. After 30 cycles of PCR, we will be going for this final elongation step at 70 to 74 degrees Celsius. So it is to make sure that if any DNA, single-stranded DNA is remaining, it should be elongated fully. Then the last step, it is final hold. This step is set up at 4 to 5 degrees, 15 degrees Celsius for an infinite time, and may be employed for short-term storage of the reaction. So a final hold is given to make sure that the DNA samples amplified is not get destroyed as till we are taking this reaction out of the equipment. So the equipment will keep the samples, that means amplified DNA fragments, at 4 degrees Celsius in a safe manner. For that only we are using this final hold. This is the schematic representation of this polymerase chain reaction, and here you can see the double-stranded DNA template or target DNA, and this pink shaded ones are the primers, forward and reverse primers, and these blue colored ones are DNTPs. nitrogenous bases. So, or nucleotides. So, the PCR consists of mainly 3 steps, that is denaturation, annealing and elongation step. And so here you can see the double-stranded DNA template, it is separated into 2 single strands after denaturation because of the disruption of this hydrogen bonds. So after denaturation, 2 single-stranded DNA are resulted. So then the second steps, in the second step, the annealing occurs, annealing of the primers to these single-stranded DNA templates. So reverse and forward primers attaches to the 2 DNA strands. And after annealing, the elongation step occurs, and here the elongation of the DNA complementary strand occurs from the primers in 5' to 3' direction. So here in the first strand, the elongation of the primers occurs in this direction, that is 5' to 3' direction, and in the complementary strand of the DNA template, here also the elongation of the primers occurs, resulting in the formation of 2 double-stranded DNA fragments. So after one cycle, we are getting for one DNA strand, one DNA template, we are getting 2 strands after a cycle, one single cycle of PCR. Then again these 1, 2, 3 steps occurs, continues, and after the second cycle, in which we started with 2 DNA strands, these 2 newly synthesized DNA templates act as the new templates for the second cycle. So after the second cycle, we are getting 4 DNA strands. From this 2 template strands, we are getting 4. Thus, the number of DNA template is, DNA fragments are amplified in multitude relationship, that is 2 raised to n. Here after first cycle, 2 raised to 1 means 2 strands we are getting, and then in the second cycle, after 2 cycle, that means 2 raised to 2, that is 4. We are getting 4 strands, 4 fragments after the 2 cycles of PCR. Likewise, we can calculate the total number of DNA fragments we are getting after a n number of PCR cycles, that is 2 raised to n. Then coming to the applications of this PCR in forensic medicine, the several fields we are using this PCR, and it is a major technique involved in almost all biological research and other fields. So first one, it is in forensic medicine, where the amplification of DNA is very useful, especially when only trace amounts of DNA is available as evidence. As evidence, if the DNA is we are getting only a little amount of DNA, so we can use this PCR to amplify the DNA template. And it is also used in paternity testing and other crime scenes also. So then selective DNA isolation. PCR, another application is selective DNA isolation. PCR allows the selective amplification of a specific region of DNA, which can be used later for many other investigations such as DNA sequencing, genetic fingerprinting, investigating evolutionary relationship among organisms. So by PCR we can amplify the selected portion of DNA. If you are getting a long sequence of DNA, then we can amplify only a selected portion of that DNA, that is a target DNA fragment. Then PCR is again used for the quantification of DNA to estimate the amount of DNA present in a sample, which is used to quantitatively determine the level of gene expression. For gene expression studies, we are using this real-time PCR is used when which measures the accumulation of DNA product after each round. In real-time PCR, we can monitor in real time. Real time, that is why it's named as real time. We can monitor the amplification of this DNA live when this PCR occurs. In the normal PCR, we can only find out the quantity of DNA only after the number of cycles. But in this real-time PCR, we can observe or we can monitor the amplification of PCR in amplification of DNA in real time, that is as live when the PCR occurs. And finally, in diagnosis of disease, the PCR technique is used. Then there are several types of PCR. So here I am highlighting some important or usually used PCR techniques in research field. That is standard PCR. In standard PCR, it is the PCR we are using in all the labs normally. That is, we use DNA as the template in standard PCR. And then RT-PCR, that is real-time PCR. So reverse transcriptase PCR. Reverse transcriptase PCR, that means we can make DNA templates from the RNA sequence, RNA fragment. So the RNA is first converted into cDNA, complementary DNA, by the reverse transcriptase enzyme, and then this cDNA is amplified using the standard PCR technique, that is RT-PCR, that reverse transcriptase PCR. Then QRT-PCR. QRT-PCR means quantitative reverse transcriptase PCR. DNA or RNA can be used and it is for the monitoring the amplification of nucleic acids or DNA in real time. Then nested PCR. In nested PCR, two sets of primers are used. Two sets. That means to be more specific to amplify this specific DNA fragment, we are using this nested PCR. Then multiplex PCR. Several pairs of primers are used in multiplex PCR. And in touchdown PCR, the annealing temperature is decreased gradually in the consecutive cycles. So hope you got an idea about this PCR techniques as a whole. So thank you for listening. Thank you.